very much, Brad. It's a pleasure to be here, and I'm sorry Don can't be here today. But other colleagues are here. Raymond Ng is here, integral to this program, Rhonda Weidman, and a few others on the team. So I'll try to give you a snapshot that will tell you where we've come from, where we are, and where we're going with our interest in making the lives of COPD patients better. And so the first concept is that COPD exacerbations, which are really like a heart attack, the lung attacks, are the leading cause of hospitalization through urgent care in Canada and in the rest of the developed world. And we spend about $1.3 billion in Canada on these hospitalizations. And if you look at this in a broader scale globally, although the detection of exacerbation is very poor around the globe because there is no test for it, there's at least 200 million people suffering from COPD, and that number's rather dramatically climbing. So one of the reasons that we were interested in this is beyond the personal suffering of patients is the economic impact that's implied with all of those hospitalizations. So we've been integrally linked to Carl Amara and his team for a number of years, and put together with the expertise in their group a number of models to look at what would be the impact of reducing lung attacks or exacerbations on costs in Canada. And even with a modest impact, 20% reduction, due to having a test that could prognose who is going to develop exacerbations, or a test that would diagnose who's going to have exacerbations, and with some slight improvements in the use of rather ineffective medicines that are available for exacerbation or for COPD management in general, we could get an $8 billion savings by the year 2025. And that's no small change. So our focus has been to develop blood tests that will allow physicians to accurately risk stratify patients for lung attacks, and also a second test to diagnose exacerbations when they actually occur and when people present in the urgent care setting, and for whom it's difficult to discriminate someone with a heart attack, with viral heart disease, pulmonary embolism, or other ailments. And since 2004, thanks to Genome Canada, we've been working to develop a biomarker development pipeline, and starting with the unmet clinical need that I've outlined for you, then embracing an approach to defining economic impact of such tests, and really clearly asking what is the question that you really want to answer in the clinic, beyond doing a market analysis to look at the possibility of wealth creation, then driving through the development of the content, the assays that have to migrate into the clinical laboratory, and then doing clinical utility assessment. All of this being done with our eye on regulatory, reimbursement, implementation, and commercialization strategies. But the main point from this slide is that it's clinic-driven. So you often hear people talk about bench to bedside, but really the most important questions, as you've heard from other speakers today, really arise in the clinic or in the community. And we've listened really hard to expert clinician work groups who've defined this program. So we started with a pilot project in 2009, before any Genome BC funding, with Don Sin in the Proof Center. And we had some early results based on patch gene whole blood transcriptome analysis, and also on plasma proteome analysis, that indicated that there might indeed be a signal for exacerbation or acute lung attacks in terms of patients who are either having them or might be more susceptible to having these frequent attacks, or as Don Sin would call them, the frequent flyers. And it was on the basis of that preliminary information and also the strength, I think, of the COPD research community in Vancouver that we were successful in putting together a program for discovery that attracted GSK. And the reason that that's important is that GSK, bless their souls, spent about $50 million putting together the best cohort to date for such studies. So they enrolled 2,800 patients. They followed them for more than 3 years. There's a number of control groups in there I won't go to. But the most important thing is they had rigorous phenotyping, as you can see on this slide. And everything that you can technically do to characterize what the nature of these patients is at baseline and then in follow-up all the way for 3 years has been done. And through the steering committee for ECLIPSE, we were able to access now about 700 participants and their samples. And they did all of the right things in terms of collecting the plasma, the PAX gene tubes, and a variety of other specimens. And it made it possible to use this as a laboratory in which to ask our question. And I'm not going to go into great detail about data today, except to show you one slide here. If you ask the question, can blood biomarkers predict which patients will become frequent exacerbators? Many patients are quiescent, have none, have 0 or 1 on a rare event. But the question that the clinicians are often asking is, who's destined to become a frequent exacerbator? And they definitely show up in urgent care and often are hospitalized. And so out of this, and this is only representing one of many, many analyses, showing you elastic net scores, that there's a group of genes, 270 in this instance, and 20 proteins that clearly differentiated patients who were going to become frequent exacerbators from those who were non-exacerbators, NE. And this was the basis, in behind this, there's a lot more analysis, but this was the basis for the funding that Brad alluded to that we received from Genome BC. And as you can see, as this program has evolved, we attracted a lot more partners who found some interest in what we were doing and wanted to get the kind of replication data and confirmation of the signatures value that we were interested in. And so we had really two major goals and 4 little subset goals here. One is the replication of the biomarkers in a much bigger cohort of patients, and the second cohort. And the second is the blood test implementation strategy. So we were going to drive this right to the clinic. And so we wanted to integrate it with what was local in COPD care, and I'll show you that, to develop a clinical reporting system that was relevant to clinicians even out in distant places in the province, and to survey to determine test choices in terms of healthcare professionals and the public, and then to develop a commercialization strategy. And so we've gone a long distance since our first preliminary results. And the essence of this slide is that if you look in terms of a prognostic test to prognose who's going to be a frequent exacerbator or a diagnostic test, we have some signals from genomic gene differential expression of genes in the whole blood or proteins in the plasma as nicely introduced a little bit ago in terms of the area under the receiver operating characteristics curve that are really pretty impressive, even to us. And the reason that they're impressive is that you see the FEV1, or forced expiratory volume, in 1 second at the bottom of the slide. Well, I just want to emphasize that there is no test in the world currently for prognosing frequent exacerbation or diagnosing them. But people have used FEV1, which is really a test for the presence of COPD, or perhaps its response to some of the therapies that are available. And it has a performance, set of performance characteristics that are really not as good as these initial signatures that I'm showing you here, derived from ECLIPSE and from a second cohort of patients in ADVAIR therapeutic trial. And so we're very, very excited about these results, and it's driven us to go further, as I'll show you in a moment. We've also been able to, with the help of the provincial government and the rapid transition program that has been set up in this province, to significantly reduce, aimed at reducing these ER visits, admissions due to acute exacerbation, and the total number of hospital bed days. We've been enrolling through this program, and we have now about 100 patients that are enrolled with all of the clinical data and samples, and it represents an additional cohort of patients for further replication. And there's also a great interest, you may know this, but two of the big chronic diseases that have the biggest impact on the health budget in this province are heart failure and COPD. And there's a province-wide integrated program called Shared Care and Practice Support Program around heart failure and COPD. And so there's a great interest in that initiative for integrating these biomarkers as part of that care once they get validated. We're also working with IO Informatics in the Bay Area, San Francisco, to develop tools to bring these to physicians and care teams, and in order to reduce this to something simple and interpretable on scoring systems that are easy, and also that have the explanation of the score level for physicians. We've done this for transplantation, and we're doing it now for COPD. And then in terms of the work with Carl O'Mara, collaboration for outcomes research and evaluation called CORE, we're determining the preferences and concerns regarding the new blood test from health administrators, health professionals, and from patients. And several focus groups have been run over the last couple of years in order to get this kind of information so that the tests really meet the need of all of the stakeholders. And then we've gained a lot of guidance from such groups as Scientia and advisors in terms of the whole regulatory, commercial, and implementation strategy in a multi-jurisdictional context. So what this has led to, something that's just been announced, is the Genome Canada large-scale applied research project that really takes this across Canada. And we have we've been able to link in both at the medical biochemistry level at National Jewish Hospital, and also in several other thought leaders in Quebec and the Cardigan cohort, in order to expand this program and get deeper replication of our results that we can be confident in and actually do the clinical utility analysis. And so this has just been recently funded, and it'll take us to 2017, when we hope that both the prognostic and the diagnostic markers will be ready for use. And then the goals of this next phase is to change the paradigm in COPD management. The deliverables are clinically validated biomarker tests for prognosis, and these would be available from primary on through tertiary care, and clinically validated biomarker tests for diagnosis, and then implementing these tools to help ensure that they're positioned for clinical uptake in the various provinces. So this, just my second last slide, this is the foundation on which we've been very fortunate. I serve on the steering committee for immunity and inflammation of the Biomarker Consortium under the FNIH, and they've just recently approved a major project with the Proof Center to advance new therapies by developing biomarkers for rate of decline in lung function in COPD, which is a very different issue than lung attacks or lung exacerbations, and is of great interest to the pharmaceutical industry that sits around the table. So we're very excited that the seed money from Genome BC is really allowing us to really grow our program, and it's really exhilarating. So I want to thank you for your attention and this time.